Welcome to lesson 4 for module 7. This is the last lesson in the module. In this lesson, we're specifically going to go over the structures and organelles that are found inside of a cell. So this might be a slightly longer lesson. There's a lot in it, but it's a good one. So learning objective here is that you'll be able to describe the different organelles, their functions, and where they are found. So animal versus plant cells. So new vocab, we have cytoplasm, cytoskeleton, centriole, cilium, flagellum, cell wall, mitochondrion, chloroplast, ribosome, nucleolus, endoplasmic reticulum, Golgi apparatus, vacuole, and lysosome. So lots of new words this time, most new organelles that we are learning about the cell. So cell structures. The environment enclosed by the plasma membrane is a semi-fluid material called cytoplasm. So you can think about the membrane like a balloon, and then the cytoplasm is this like gel-like stuff that's filling it. So in prokaryotes, all of the chemical processes of the cell take place directly in the cytoplasm. Because remember, prokaryotes do not have organelles at all. So they don't have these structures for things to happen in. So it's just going to happen in that space. In eukaryotes, these processes take place within organelles in the cytoplasm. Most do. So the cytoskeleton is a supporting network of long, thin protein fibers that form a framework for the cell. Microtubules are long, hollow protein cylinders that form a rigid skeleton for the cell and help move substances within the cell. Microfilaments are thin protein threads that help give the cell shape and enable it to move. And centrioles, which are made of microtubules, function during cell division. So cilia and flagella are made of microtubules, and all of these structures in Module 9, when we talk about cell division, are going to come in handy to know what they are, what they do, because they are helpful there too. So cilia are short, numerous projections that look like hair and then move in tandem like oars in a rowboat. Flagella are longer and less numerous than cilia. They move with a whip-like motion. So flagella usually looks like a tail at the end of a cell, whereas cilia are like a hair-like thing going all around the outside. All right, so cell structures here. Move myself up here. So these are cell walls, these structured things on the outside. It's a thick, rigid mesh of fibers that surrounds the outside of the plasma membrane, and the cell wall protects the cell and gives it structure, and it is made from a carbohydrate called cellulose. The cell wall of a prokaryote functions similarly to a plant cell wall. The prokaryote cell wall protects the cell and gives it structure, is made from peptoglycan. And then for controlling cell activities, we have the nucleus. So the nucleus of a cell contains most of the cell's DNA, which stores information used to make proteins that determine a cell's growth, function, and reproduction. All of the instructions are in the nucleus. So the nucleus is surrounded by a double membrane called a nuclear envelope. It has its own little cellular membrane. For energy, we have the mitochondria. That would be the plural. For a singular, it's mitochondrion. They convert fuel particles into usable energy, and they have their own things going on in here. They have an outer membrane and then a highly folded inner membrane. So the outer membrane is red here, inner membrane is yellow. That folded membrane provides a lot of surface area for the breaking of bonds in sugar molecules. Because if you remember, for cellular respiration, that's where this happens. And then for processing energy, we have chloroplasts. So plant cells and some other eukaryotes will contain chloroplasts, which capture light and convert it into chemical energy in a process called photosynthesis. Chloroplasts are composed of multiple small discs called thylakoids, which contain the light-capturing pigment called chlorophyll. So it's these little thylakoids down here that absorb the light and help photosynthesis happen. So for manufacturing proteins, ribosomes are organelles that manufacture proteins. So there are these little dots right here. Unlike other organelles, ribosomes are not membrane bound. And they are produced inside of the nucleus in the nucleolus. So this is the nucleus right here. The nucleolus is in here. So this is where ribosomes come from. And then outside of the nucleus, which is right here in A, we have the endoplasmic reticulum. So that's a membrane system of folded sacs and interconnected channels that serves as the site for protein and lipid synthesis. So it's rough, there's rough and smooth. Rough right here is going to have the ribosomes on it, and then smooth has no ribosomes. So the Golgi apparatus is a flattened stack of membranes that modifies, sorts, and packages proteins. Proteins are packaged into sacs called vesicles, which can fuse to the plasma membrane and release the proteins to the environment. A vacuole is a membrane-bound sac used for temporary storage. Vacuoles store food, enzymes, or waste. They can be found in both animal and plant cells. In animal cells, they are usually temporary. In plant cells, they are always there. They take up a lot of space. So processing, transporting, and storing molecules. Lysosomes are vesicles that contain substances that digest excess or worn out organelles and food particles. So they break everything down. They also digest bacteria or viruses that invade a cell. So if it's a white blood cell, they're going to have a lot of lysosomes because they need to be able to break down all of that bacteria and viruses. All right, so summary and practice questions. So this is an animal cell. Here are all of the different organelles that are found. Key difference we will see between animal cells and plant cells. Animal cells do not have that cell wall, and they do not have chloroplast. Plant cells, we got a very structured cell wall. We still have that cell membrane on the inside, and we have a chloroplast right here, and then a big old vacuole. Prokaryote cell, so here we have the, this would be like the cilia, and then this would be a flagella. There is no organelles here. It's just a big loop of DNA with cytoplasm. So plant cells have chloroplasts and chlorophyll, vacuoles, cell walls. Animals generally do not have these features. They very rarely would have a cell wall, sometimes have vacuoles, and very rarely would have chloroplasts or chlorophyll. So organelles at work. Knowledge of organelles allows for an increased understanding of cellular processes. Protein synthesis begins in the nucleus with DNA. RNA and ribosomes leave the nucleus and produce a protein on the endoplasmic reticulum. Proteins packaged and produced in the endoplasmic reticulum are sent to the Golgi apparatus for packaging. Packaged proteins are delivered to other organelles where they serve a variety of functions. All right, review question time. So first one is, which structure is found in both plant and animal cells? Always. Not just sometimes, but always. Not chloroplast, that would be a plant. Most vacuoles are in plants, and not a cell wall, that is plants only. It would be a ribosome, are found in both. In which structure would you expect to find a cell wall? Human skin cell, leaf from a tree, blood cell from a cat, liver from a mouse? If they're only found in plants, we would expect that from a tree. Which structure synthesizes proteins that will be used by the cell? Where are we getting those proteins from? It's the ribosome. Ribosomes come from the nucleolus, and they are on the endoplasmic reticulum, but the actual synthesis happens in the ribosome. Which is not a component of the cytoskeleton? Not the microtubule, or yes, the microtubule, yes, the centriole, and yes, the microfilament, just not the cell wall. All of these are part of the cytoskeleton, cell wall is not. Which is a site for protein synthesis? Not the nuclear pore, not the nucleolus, chromatin. So it's going to be the endoplasmic reticulum. So remember, the ribosomes that are on the endoplasmic reticulum are where the actual synthesis is taking place. But out of these options, endoplasmic reticulum is the location. All right, that is it for that last lesson. There are additional resources posted on Canvas to dig deeper into each of these organelles. But if you have any questions, please let me know, and I will see you next time.